points, and if we plot their intensity, they will be far separated. Okay. Now, so they have separated image points. Now slowly we bring those points closer and closer. Okay, we are taking them closer and closer. So there would be one particular position where the minima of one would be coinciding with the maximum of the other. Okay, and that point is where our eye can separate these 2 points, and that criteria is known as Rayleigh criteria. Okay, and typically the length would be 0.61 lambda by N, and Na is numerical aperture. And next, when we have taken the point still further, okay, and we have a graph. So if we are continuously superposing, there will be a point where they will have a dip, and there would be a final point where this dip will be just crossover. That point is known as Sparrow criteria. That means still previously you could have had scanned a semiconductor detector and still would have seen 2 points. Okay, but in this particular case, after this it cannot be done. This is very important because when we talk about metrology, we have to separate 2 points, and those 2 points separated most of the time. Now there is a semiconductor detector, so therefore that semiconductor detector will be essentially scanning the sample. So that has to set the resolution criteria. Now this is a conventional microscope. All of you have seen it, I think. So here we have an object, then there is an objective lens, we have a tube lens, and then we, from an eyepiece, we view the object. Okay. Here also, yeah. So here also, if we are trying to find differences between 2 points, we see that from the peak, this bottom value is about 70%, 0.73. Okay. Now, but the problem with normal microscope is there are many lights, stray lights, which are coming into the picture, and that actually blurs the actual image. So now there is a confocal microscope which essentially separates the 2 lights. So typically, in case of confocal microscope, in conventional microscope there is no pinhole along the source. Okay. Here we have a pinhole across the source and also the detector, and that will essentially cut off the light. How much time I have? So in case of confocal microscope, you can actually go into a depth resolution of 10 nanometers. Okay. And a lateral resolution, that means XY resolution, which is the surface, of the order of micrometer, and we'll just going to see that. So this is how